All right, welcome.
Thank you.
So Barry, the point of this, as we've sort of chatted about, is I want to try and walk people through, or even myself really, walk through a bit of how did we get to where we are now in terms of your most recent work, particularly ultimately getting to the work you've done around the Glycocalyx and endothelial surface layer and the implications of that from a biological perspective. But so that we can understand that, I think it's really useful, it's certainly been very useful for me to kind of understand how you've got to that point from a thinking perspective, right? So starting from your early work and what sort of led you to that scenario, because I find in trying to explain some of these terms with people, it's very difficult for people to understand, what it is you're talking about if they're not coming through the right lens, if that makes sense. So how about we start with, you've got, a very comprehensive background, so we'll try and make this reasonably short and concise, but I want to focus on particularly your early work and try and walk people through, your career around, the early work you did from a graduate and your PhD, etc., and how that ultimately led you to where you got to from a self-assembly sort of scientific perspective. So how about we start from the beginning? Your first early work from an academic point of view, what were you doing?
Graduate work?
Yeah.
Oh, okay. I finished my master's degree in the University of Western Australia and then went to the United States to do a PhD. And I was a mathematical physicist. But I was also a biology tragic, because I really wanted to do chemistry and biology, but I couldn't because the timetable was such that you couldn't. You had to do physical sciences or biological. So I became a mathematical physicist because that was easy for me and I had the skills. And I went to the United States and became a mathematical physicist and ended up finishing a PhD at IBM Research in New York, their new lab, actually, the new computer lab. So that's how I started, and I came back to Australia for 7 years and was in applied mathematics and still did physics, but then had the opportunity to go back to the States and start doing some biology. And I went back to the National Institutes of Health, which is the big centre in the United States, where most of the medical research is controlled from. And I teamed up luckily with a friend called Adrian Parsik, and he was a biophysicist, but he was stuck on a big problem on molecular forces, how lipid molecules, the molecules that form cell membranes, interact, and what are the forces between them, and how do you calculate those forces? And it was a time when a big breakthrough in mathematical physics had come through to deal with molecular forces. It was the tip of the iceberg, and quantum mechanics was in full flight. And there was a theory developed by some Russians, mainly Lifshitz, the main guy, and they made this beautiful theory and really sophisticated quantum mechanics, the creme de la creme, but nobody could use it. It was too complicated, and it was just this beautiful, sophisticated theory. So Adrian had this idea who was going to use the theory and put it into biology and make it work, and he was stuck on making a theory for one layer of these molecules, or many molecules, many layers, and I said, well, why don't we just do one layer instead of an infinite layer? So we did, and it worked, and he worked out how to put in all the different properties of different materials, oil and water and proteins, and we found out how to make the theory work, and I worked out all the mathematics, and so we made a big splash, and we're quite popular. And then I came back after a year or two to Australia and was given a chair at the Institute of Advanced Studies That was like the Harvard of the Southern Hemisphere at that time. So I had this chair and could do what I liked. Not many resources, but so I built this department to do exactly that, to take physics and physical chemistry and to put it into biology and to put it into chemical engineering as well, because the same language, the field of physical chemistry, colloid chemistry, molecular forces, is the enabling discipline in principle behind all of molecular biology and behind all of chemical engineering. And the theory was very primitive, and we got going and made it more and more sophisticated and started to put it to use, to do something useful. So that's how I started. And I had a department for, now I founded in 1970.
So this is now at ANU. You've
At
started
ANU.
that department at ANU. Yep.
Department, and I was able to recruit a huge number of really brilliant young people. It was a wonderful place to be. I was very lucky. And we recruited heaps and heaps. They were all very bright, and they all had the same idea. They were interested. It was the time after the molecular biology revolution, when everybody was doing molecular biology and everybody in medicine was, and it was the big go. So lots of physicists and mathematicians wanted to get into biology. It was exciting. So I was able to recruit a huge number of very bright people, and we became the world centre in this field. And we learnt more than molecular forces. We started to measure them. We were the first to measure molecular forces since Isaac Newton, very successfully, and we developed theories of how molecules interact through these molecular forces, whose subtleties we understood by now, and how they got together and assembled into things like cell membranes or into smaller organelles that were purpose-built in biology, and nerve membranes and all kinds of things in biology. So we were the leaders, the beginners in that, quantifying that field from the physical point of view. So that's how it happened.
Okay, so you sort of come at this. Well, there's a couple of key things I think. Is first of all, you've come from this from a sort of, a base on understanding forces and how forces, interact, right? So very much a physics scenario as opposed to, the way I kind of, when I did my sort of science through high school into university, you had this kind of very clear delineations. Physics was around forces and Newton's laws and Einstein's theories and those sorts of scenarios. And then when you moved into chemistry was all electrostatics and mixing chemicals and bonds and those sorts of things. And then biology was very much at the sort of macro scale of, cells and tissues and organs and that sort of stuff. You've come from the very foundational, if you like, in terms of not just trying to understand, how these things form in the first place, but particularly from a measurement point of view, in terms of what can you demonstrate when in certain environments, how do these things self-assemble? Would that be fair to say?
Yeah, it's being able to, a proper theory, you have to predict what's going on. You make a theory and you have several parameters like the length of a chain of oil for a molecule, a lipid molecule, which is like an oil molecule that forms biological cell membranes. It's got two oil-like molecules and a head group that likes to be in water. And how these interact, just molecules floating in water, and form particular structures, you want to be able to predict what kind of structures form, depending on the oil, the temperature, the chain length, the head group, the salt, and so on. And so we built these theories based in something called statistical mechanics, the fundamentals of thermodynamics at the molecular level. And so we're able to predict what structures form with different oils, different surface active agents, different proteins, and to do that predictably and then to apply it to biology. Because once you understand how if you change the chain length of a lipid, the whole structure will change, then you can predict the whole thing. You can make things work. Well, you can't predict the whole thing, but you can understand how a small change in a molecule, like a drug, can make a big change in the whole physiology.
In terms of its ultimate effect in that environment, yeah. So, okay, so if we fast forward, so you've been doing this for a few decades now, you've got collaborations now with people really all around the world, not just because of who sort of come through the department, but also your collaborations, particularly through Europe and the US. So we've gone through this sort of self-assembly scenario, and then if we sort of jumped to sort of 2018-19, I think, where you did your first work with Brandon, sort of trying to understand the endothelial surface layer itself, that was really the catalyst for the most recent deep dive into biology?
Yes, we spent many years developing theories of relevant to cell biology, involving these molecules called lipids and water and oil and so on, how they assembled into different structures. But the big change, the add-on to that, was when we started to extend that work to include it, to involve polymers.
Okay.
And polymers are another class of molecules besides proteins. Proteins are wound up polymers all tied together in blobs. But long, thin polymers are another class of molecules besides the ones I mentioned: oil, water, proteins, lipids, and surface active agents. And there the structure that form were just completely unknown. They were much more complicated, or they seemed to be much more complicated, and they're unknown. And the junction with biology, the further extension to biology into to make it a much wider and more broader application, was when we started to deal with polymers, because we'd dealt with cells up to that stage and everything related to cells and interactions between colloidal particles. But in physiology, you have a whole class of structures that involve these polymers. You have, for example, a nerve, which is built of lipids, a tube, but it's covered with this polymeric layer. And the cells are covered with polymeric layers too. And they're called the Glycocalyx and the endothelial surface layer, and they're made up of polymers. But for biologists, they were just blobs, blurry, nothing material. It was just a material that didn't matter. Whereas all the attention was on the cell membrane and how the cell membrane conducted ions in and out with ion pumps and all kinds of complicated simulation. But this fuzzy layer, the fuzzy layer was just something that was a fuzzy layer. But the fuzzy layer was huge. The membrane of cells is only about 3 nanometers thick. It's about this much. But the glycocalyx is about this much, made up of polymers. They're things called heparin sulfate and chondroitin sulfate. You can buy them at the chemist shop. And then there's a thing called the endothelial surface layer, which is much sparser and very thin, and it stretches out for 1 million miles, a micron. So this was just fuzz, nothing. And all the attention was on the inside of the cell, on the DNA, on the ion pumps and so on. So this was a completely new area, and obviously a very big area because the stuff is so big. So we started on that just about 7 or 8 years ago.
Yeah. So I find that fascinating. We'll come back to that because that's really where the sort of meat of the discussion I think we want to ultimately get to. But I find it's a very difficult thing to understand in relative scale, I think, because when you look at these things under the microscope or whatever sort of instrumentation allows you to actually give some sort of imaging or of these things, I find it, and maybe it's just me or maybe it's a more natural sort of human thing, it's very difficult to get a full appreciation for scale. And so when you talk about that, the analogy I've sort of come to understand is almost like a rugby field, right? And so the rugby field, you've got this 100 metre long field and you've got this sort of 10 metre in goal area, and then you've got the line, the dead ball line at the very back of the field, that's the cell membrane. That's the 3 nanometers sort of thick scenario, and you've got to pass to get into the cell. But you've got to get through that dead ball. You've got to get through the in-goal area first. That that glycocalyx, right? And that's this mesh of all these different polymers of with quite a degree of complexity. And then you've got the entire field to get through, which is where you have that very little organic matter, very little. It's this big sort of exclusion zone with some sort of branches and stuff sort of heading out to it. But you've got to run the gauntlet of the entire field of the ESL. You've then got to get through the in-goal area to even get to that dead ball line, and yet all the attention is on that sort of dead ball line. And I find that understanding that degree of scale, helps me sort of appreciate that there's a lot more to this sort of story than just that line, which is it's important because that's the edge of the boundary, but there's a whole game in front of that. So I just want to go back a step though, because I think one of the things in terms of how have you come to the understanding, I suppose, and you often talk about the 4 elements and how that they've been incorporated into particularly physical chemistry, etc. And you talk about the fact that in particular the role of dissolved gas. So do you want to sort of walk through the progression of that thinking?
Yeah. Well, the development of the theory of physical chemistry, it's a venerable theory. As I said, it's an enabling discipline behind a mess of lots of other disciplines, biology, botany, chemical engineering and so on. But it developed as a theory with electrochemistry, which is all the go now with electric cars and electric windmills and so on, about 1900, after Faraday, through the 19th century. And it took form as a mathematical theory, if you like, around about 1900 and 1910. And the first 8 Nobel Prize winners were all in electrochemistry.
Well, this is where electricity has been invented, if you like, and is actually starting to be, so that makes sense.
It's salt in water, batteries and so on. So that's when it started to take off, and that became the foundations of physical chemistry. There's more than that. But then along came quantum mechanics in the 1920s, and that was a huge change. In fact, Planck quantum mechanics was 1900. So it changed things completely, because before that one thought about charged molecules, proteins or salt molecules, just in terms of electrostatic interactions. And then along came quantum mechanics, and there's a new kind of field added infinite diversity to the whole thing. And interactions became much more specific, whereas before, if you had in a solution, a salt solution with sodium and chloride, sodium is positively charged, chloride negatively charged. You had potassium chloride, potassium is positively charged, chloride negatively charged. You'd accommodate the differences in their interactions just with changing the radius, sodium ion or potassium ion. But you could have a different size, but that's just fitting. And then it turns out when you add, that was a whole missing elephant in the room in physical chemistry. You could not understand specific ion effects. So if you've got a theory about ions in water and you say everything's specific, then the theory doesn't work. Not a good theory. But anyway, that
always works. Right?
No, it always works.
Right?
And all of electrochemistry,
not predictive, but it's always
all modern batteries that we're using for climate change and so on, the electrochemistry based on a theory that doesn't work. Very good, but a bit of a gamble. So anyway, quantum mechanics changed all that, and so the forces between neutral atoms, like argon or helium and so on, became explained. The forces between ions, extra forces, specific things became explained. So that all changed the theory. And that was one big advance. But then the other thing that happened, the foundations were then laid even more, foundations of things like pH and buffers and surface tension, concepts we deal with all the time, all fine. However, it didn't work because the theory was fundamentally wrong. It was wrong because they dealt with the electrostatics in one way, a non-linear theory, and the quantum mechanical van der Waals forces in a linear theory. So fundamentally it's wrong. So you keep using it, and then came the killer, where all of our recent work came from. We realised that that was only half of the matter. The real problem was that we'd forgotten about dissolved gas, and reasonably enough, because the amount of dissolved gas in this water glass here is tiny. It's nothing. It wouldn't make any difference. So okay, we forget about that.
But it's also constant, right? In the sense that, you know, in the real world,
oh, it's always there.
There's always some dissolved gas of some amount.
Yeah. Yeah. So you've got a theory that says, okay, we've got all this sophisticated quantum mechanics. Yeah. We've got all this fancy electrostatic theories, electrodynamic theories, and admittedly we've buggered it up because it's inconsistent. But nevertheless, that's it. However, we've forgotten that in the real world we have all this dissolved gas. Tiny bit, but it turns out the dissolved gas makes a huge difference. The dissolved gas isn't just gas, it's organised into little things called nanobubbles that are bundles of energy and got lots of energy that drive reactions. So the whole thing is a mess. And since the biologists and the medicos use the theory, which doesn't work, they don't get much joy out of the theory. And so they turned to something called computers. And computers decided they would do the problem. They could simulate all manner of things because they could put in all kinds of...
So you could do the experiments now mathematically as opposed to doing them physically, and therefore you could well, first of all, the cost of research drops drastically, right? Because you can now run... Well, the computers got very expensive, but they're still much faster and can run way more different experiments because it's...
I make heaps and heaps, and they're all completely meaningless because they're modelling something that isn't. They have a model on the computer that puts in molecular size and interactions and so on, and temperature, but it doesn't deal with the fact, and it models a system which doesn't have dissolved gas. But everything in the world does have dissolved gas, and when you've got dissolved gas there, it's upside down, completely different to if you don't have dissolved gas. So they're modelling something and get a perfect fit.
So would it be at least, you know, maybe charitable even, but it would seem reasonable on first glance to me that if you've got essentially, and going back to my simplistic, you know, background in science, is if you have a constant, science is all about understanding the impact of variables to a large degree, right? So if you have a constant, there is always dissolved gas. It is always roughly the same amount at a given temperature and pressure and all those sorts of things. You could at least understand to some degree that, okay, if it's always there, then it's not a variable we need to consider. So,
oh, it's only a tiny amount.
And it's a tiny amount, that's right. So when tiny amounts without no real variation, et cetera, you can kind of ignore it in the sense of like, because it's always going to be there. I guess, you know, what it seems to me like you're saying is it may always be there, but the amount and the type does matter because small changes in the conditions can change the effect that has. You often talk about the removal of the dissolved gas and the effect that has.
Yeah, if you remove the gas, everything changes. There's a thing called hydrophobic interactions, which describe oil in water.
Everyone knows that oil and water don't mix.
Oil and water don't mix.
Yep.
So and that's called a hydrophobic interaction, and something that mixes with water is called hydrophilic. They're anthropomorphic words that don't mean anything, except that's what it is.
Explain a phenomena that's observed, right? So if not that I have any, but if I was to pour some oil in here, the oil is going to form a layer on top of the water, and they're not going to mix, right?
Yeah. So if you take the gas away, you degas it, the hydrophobic interaction goes away, and you can shake up oil and water, and have a suspension of droplets of oil and water, and they're stable. They don't demix. So hydrophobic interactions depend on dissolved gas.
A function of the gas, yeah. Yeah, and because we don't observe that in the real world, because we always deal with what, yeah,
and also we breathe.
We do,
and that's a function of life. If you don't breathe, you're going to have
well, it's
a really
like not I
hard
think,
time.
therefore I am. It's I breathe, therefore I can think. Yes, so I have the option of thinking. But again, sort of, it's not unreasonable in terms of progressing of thinking to think, well, if it is always there and there's always going to be degrees of that, I guess. That's fine at the sort of macro level to some degree, because it's virtually impossible to have a gasless water, if you like, in any sort of large sense. But when we get down to the scale of particularly microbiology and that sort of nanoscale, then the amount of dissolved gas can vary dramatically in relatively small areas, right? Even though you're not going to degas your entire body, but there can be different areas of cells or intracellular spaces where that can match.
The gas separates out into little areas in salt solutions or sugar solutions or amino acid protein solution. It separates out segregates out into little containers called nanobubbles, and they're stable and they're there.
So let's get into that, because the stability of bubbles and the bubble fusion inhibition is, I think, again, one of those really sort of fundamental building blocks that people don't fully understand. So when we're talking about dissolved gas, we are talking about the molecules of oxygen or nitrogen or whatever in water, and the forming of nanobubbles. Can we just talk about that bubble-bubble fusion interaction and why and how we can know that these nanobubbles do exist despite the fact that they're so small and virtually impossible to see?
Well, it all started with work that well, it's been known for a long time, since the turn of the century, that if you take bubbles, you bubble like a fish tank. You bubble air through a frit and you have bubbles, and the bubbles go up a column, and you do that in your fish tank, then you see the column is clear and the little bubbles combine to form big bubbles. They fuse and become big bubbles and they go up to the top. But if you add salt to the thing, then at a certain concentration, which is exactly the salt concentration of salt in our bodies, suddenly the bubbles coming out of the frit don't fuse, and it turns into a whole whooshy.
It's cloudy essentially, because those bubbles.
Little bubbles, dense foam and so on. Well, this has been known for a while and forgotten. It was used by Russian scientists to float coal. You have these little bubbles and the colloidal particles stick to them and they float to the top. And separates that from the soil. So I opened an international conference in 1980, which we had in Canberra, of 2 international unions, and I started with a demonstration of this thing. And these are all eminent colloid chemists around the world. And they said, okay, well, the theory will explain it, we don't know. But then didn't explain it. And then it turns out if you use different salt particles, like sodium acetate instead of sodium chloride, if you add salt, nothing happens. The bubbles continue to fuse. So there's some salts, we have this phenomenon, bubbles are inhibited to fusing, and others that don't. Well, this is utterly unexplained by any of these theories that we have got to so far then in the 1980s. And it's still not explained. And it's mysterious because it's the simplest experiment you can think of in the world. If you look at the ocean, it's foamy due to this effect. There's lots of salt in the ocean. If you look at a fresh lake, you look at the bubbles blown by the wind, they instantly fuse and dissipate. So it's a fundamental problem. It's very simple. But then so you think, okay, well, you can explain it with our theories of electrostatics, but you can't, and you can't explain the specificity. And then it turns out they have the same effect with sugars. glucose and fructose and sugar. We use all these different...
So just before we go on to those, if we go back to that, so it's just to be clear, so from a salt perspective, from an anion, cation sort of, so we can demonstrate which ones do and which ones don't, but just to clarify, I think what you're referring to is, you know, I can change the anion and cation, and there's no consistency around sodium will always be there in, sometimes it is, sometimes it isn't, right? So sodium chloride, you gave that example, where sodium acetate not. So we've got this sort of like table, if you like, of these combinations do, these combinations don't, but there's no obvious consistency across the...
No, well, I mean, there's a set of rules that tell you which pairs are going to work.
Yes.
You can assign a flavour to each positively charged ion, a cation, or each negatively charged ion, which is an anion, and you look at these rules. There's combining rules that work, just like the periodic table, but nobody knows why. But then it looks as though you can make theory, but you can't. And then it turns out the same thing happens with sugars. There's sugars that exhibit this effect, stop bubbles from fusing, and there's others that don't. Glucose doesn't, and fructose does. So there's this and then there's a strange effect with amino acids that are involved with proteins. Some work to stop bubbles forming, and others don't. So it's still a mystery, but there's more and more evidence that shows that they really are there, and that nanobubbles are tiny bubbles that you can't see. They're of the order of 3 nanometers thick, tiny.
So just to clarify, because that's again something often sort of misunderstood, people when they often talk about nano, they can often be talking about more of a sort of micro sort of scale, right? So nano, we're down to sort of single double-digit nanometers essentially, not into the sort of you know 100.
That's right. Yeah, most people talk about. There's a great interest in nanobubbles these days, but what they mean by nanobubbles are microbubbles, great big bubbles, and for microbubbles you can apply macroscopic theory. Temperature, pressure and so on, but you can't for nanobubbles. It's too tiny, they're too small. And these nanobubbles are involved in enzymatic reactions, and they drive all chemical reactions, and people haven't known that. So that's a really big change, that you've got something that's creating reactive oxygen species, nitric oxide, free radicals, all kinds of active species inside them, and those things drive chemical reactions.
Okay, so
that's a big change.
Yeah, so we want to get into some of that effect. So I just want to go back to the salts versus sugars versus amino acids thing, because I think that level of detail is super interesting as well, in the sense that because you know you've got charged particles in terms of salts, you've then got sugars, and then you've got amino acids, which are fundamentally very different molecules, not just in size and shape, but also in charge. But the other interesting thing around that, not just that they all seem to have this sort of critical concentration where if they have an effect, it seems to be at a relatively narrow band of transition of critical contribution, but particularly with the amino acids, that the work on that showed that the nature of the bubble size can vary dramatically as well as concentration, right? So a certain amino acid, you know, not only will it form, its critical concentration will be a fraction of a different amino acid's critical concentration, but then therefore the nature of the size of the bubbles naturally form, say if we're doing an experiment adding say tyrosine versus, glutamine or whatever, experiment. So you would talk a little bit about why and how that occurs, sugars versus amino acids and sort of bubble sizes. What do you think's going on there, or what we can demonstrate those things? What can we speculate around the reasons why it happened, or what can we dismiss as can't be the reason because of those various interactions?
Well, it can't be just electrostatic forces, which is the obvious thing you think of, because these other molecules, sugars and amino acids, are not charged. So there's no charge involved, but still there's gas bubbles going to the surface, nucleating at the surface and cavitating and forming little bubbles. That's one way to produce them. The other way to produce them is just, say, in salt or other solutions. They exist by themselves. They're just hanging around, existing. And the nanobubbles are the source of the force that stops big bubbles coming together. I mean, you've got fusion inhibition, say with sodium chloride, at the same concentration of salt as in our bodies, so there for some reason. And for other salts, sodium acetate, there's no fusion. Well, there's a force that we've measured and studied a great deal in theory, in liquids, which is called a depletion interaction. And that's due to small, not with nanobubbles, with molecules that are quite large, of the size of nanobubbles. So if you measure the forces between two surfaces in which you've got these molecules, you get these forces called depletion forces, where they oscillate up and down as you come together. They're no longer an attractive force coming to two charged surfaces. They oscillate up and down, and you've got to push over those oscillations as you bring them together. So that provides the source of a repulsive force that stops them coming together.
And so eventually, if there's enough concentration of this sodium chloride in this example from a bubble scenario, if there's enough in there, then those forces, there's too much of a hump to get over to continue to get closer together.
So you get these nanobubbles forming. And they're the same, divert a bit, they're exactly the same as exists for something called surfactants. Surfactants are surface active agents, as I mentioned before, a soap, like olive oil with a head group that's negatively charged, and they form micelles. Micelles are little nanostructures with all the tails sticking in and all the head groups together. They assemble at a critical micelle concentration. They're just like a nanobubble and the same size. Okay, and so you get the same. You can measure the same force.
So sorry, just to clarify, because this is a really important point, I think. So if I have a solution and I'm continuing to add surfactant to that solution, then they're all sort of floating around, because there's not enough of them. But eventually I get to that critical concentration, and once that happens, they can now aggregate and form these structures.
They form these structures at the same size as the nanobubbles we're talking about. Yep. And this is reflected in, if you measure conductivity with charged surfactant. You measure the conductivity, it's going along, and suddenly there's a break because the structure of the solution's changed. It had monomers going along, charged, but then suddenly they're together, and so you get a break in the conductivity. The same thing happens.
So you can actually measure that critical concentration.
Yeah, and so you can do the same with dissolved gas. If you measure, you're measuring the conductivity of a salt solution, and you change the salt solution at a critical concentration, you'll get a break corresponding to the formation of dissolved gas nanobubbles. So it's just like surfactants. These things aggregate, the same as surfactants aggregate, the same as all molecules aggregate. And it turns out they can aggregate at a surface, at a hydrophobic surface, or they can form spontaneously in solution. There's both kinds, and they're responsible for something called cavitation. It's been known for many, many years that if you try to break water apart, if you have a propeller that drives it, you get a loud noise from breaking the water apart. If you take away the dissolved gas, you don't get cavitation. And the strength of water is determined by these dissolved gas particles or nanobubbles. And if you take them away, the strength of water is 100 times greater than if those defects are there. So they're there for sure.
So just to clarify, so you wouldn't want to be diving into a pool of degassed water if that was possible, because you'd be smashing your head against the. So not just breaking up the surface tension like they do with the diving, not just that, the actual tensile strength of water.
No, I don't know if anyone's done the experiment.
A degassed environment, you're not going to last too long, but yeah. So it's really interesting because cavitation is one of those very interesting things that's, I guess, well understood by anyone, particularly anyone who's had any, experience boating, right? And the problem with cavitation is, if the propeller spins too fast, essentially, and cavitation occurs, you get these bubbles. And yes, it's very noisy, and it reduces the effect of the propeller. But the effect of cavitation, if you look at what happens to a propeller blade that's been subject to cavitation, it looks as if it's actually been corroded, right? It's not a, and therein lies a bit of the hint, I suppose, in the sense that this isn't a physical force in the sense of, you know, you're sandblasting, you know, the propeller. You're actually corroding it as if it's been dipped in sort of acid, essentially, yeah? And that's the effect of these nanobubbles.
Yeah. Exactly, and it costs a huge amount of energy to do cavitation. If you take away dissolved gas, it's much cheaper to run a propeller. But that's the same thing that drives the energy of enzymes, these nanobubbles. The picture of biologists of enzymes is that they have an active site in the molecule like that, a particular site, and along comes something you want to cup up, and there's an enzyme and then a substrate. You want to chop it up, it comes along and then it chops it up. But it does it by, the enzyme is hydrophobic. It's got a metal ion of a particular kind there that sets the structure. These are different amino acids in the protein, which is the enzyme. And along comes the substrate. I'll make it this one. And under the influence of molecular forces, it comes along, comes up to this thing, chomps into it. That creates the cavitation.
So again, just to clarify, so this is your two surfaces sort of coming together that are both hydrophobic, and so at some stage...
No, they're not both hydrophobic. Only one's enough.
Side, yep. So the inside of the enzyme essentially is hydrophobic. Yep. So by hydrophobic, what we mean from that is it's trying to push the water away for...
That's right. So when it comes,
and if that water's got nowhere to go, because this thing's now sort of closed, essentially we've got a sort of packing problem, right? We're trying to shove things into...
Well, the water gets pushed out. And as it does, you form this tiny little nanobubble inside. And
just to clarify, and this might be, you know, in my mind, what I'm sort of seeing here is as this thing sort of coming in and it's pushing the water out, as you say, something's got to replace the water, right? And that's why this nanobubble's forming spontaneously, if you like. The gas is aggregating external to the water, if you like, in this area as a result of this
in that hydrophobic region. And as it does so, it's a spontaneous mini-phase transition. The water's kicked out, right? And that requires energy. And that energy rips apart water and nitrogen and oxygen inside the nanobubble. It's a way that I think of as nature has to harness all the weak molecular forces we talked about, van der Waals forces that drive the two together. Harnessing all those collectively, that's a phase transition, and boom, so that energy is enough to, in what's called a restriction enzyme, to cut a phosphate bond, which is a large energy, so you do it cooperatively. And those species inside, reactive oxygen species, hydrogen peroxide, nitric oxide and so on, they're all these reactive species that biologists know are there, and they're created by the gas
by this physical interaction. Yeah. So you know, I've heard you talk about this a lot, and so my macro analogy to this, I see this as almost like a lightning strike, right? Is it because again, you know, my training in terms of chemistry and biology sort of got to, you always had these magical things called enzymes, and you knew enzymes were critical because if you take enzymes out, reactions don't occur, or they occur so slowly that they can't explain what's sort of going on. So my training was always enzymes are catalysts that reduce the energy required.
That's right.
Which is a slight thermodynamic problem. Interestingly, the same teacher who taught me biology taught me physics, so I'm not sure how he combined the things, but so you had this sort of these magical enzymes were really just proteins folded up in a very specific way. If they don't fold that way, they don't work. And if they're not there, the reaction doesn't work, or at least doesn't work in anything like the speed, if you like. So, but no one ever seemed to have an explanation of how do they break the law of thermodynamics, or how do they harness all this energy? And to me, what you're describing is similar to a lightning strike, where you have all of these, you know, charges around that suddenly coalesce into one space, and we get this enormous amount of energy release at a specific place. Is that a reasonable way to think of it?
Yeah, well, the biologists don't have that problem. Physicists have a problem because they are interested in energy. Biologists solve that problem very easily. They have a word for a molecule, Arthur, call it Martha. Now, Arthur's doesn't do anything. But if you put a hyphen and ASE, ACE, Arthurase, it's an enzyme. And then it's energized. It's energized by the postfix.
Like folding into the right shape
no,
and adding the
it's all
ACE.
folded. No, you add ACE to it.
Yeah, well, you've got to fold in the right shape, then you're a working protein, right?
Yeah, but it's not going to work unless you add ACE to it.
No, that's right. And then you just have to add the ACE to it, and that gives it its superpower. That's Superman's,
right.
He's got to be on the yellow sun.
And it's no problem if you're not interested in energy. We don't know about energy.
Yeah.
In the same way.
The energy, but in biological processes, energy comes from ATP routinely, right? So we're not talking about the chemical energy release or whatever. We're talking about I guess there's a degree of even thermal energy, I suppose, involved in this. This is a result of forces being pushed to an extreme, if you like, what you're saying, and therefore something's got to give, right?
Yeah, but biologists don't think in terms of forces at all. They think about binding
and electrostatic.
No, they don't think about electrostatic. They just think of binding.
Right.
They don't think about forces at all. But
but to bind, one has to have a physical force, right? Because I guess when you get down to that kind of micro level, I mean, even if you were to take the smoothest surface possible and you run something along that, there's no such thing as a molecularly smooth surface, consistently in nature, sort of.
They don't think about it. They have a computer program that does that.
Right. Okay. Right. So I guess peeling back then, because you have to answer this in the sense of what forces are involved and how will those forces behave, and particularly because you have this history of understanding what those forces are, being able to demonstrate through measurement in terms of in different environments what forces are at play, and building models that would then allow you to predict, experimental outcomes based on those things. Whenever you're faced with an environment that contains all this, then those answers have to be. You have to have an answer to what would happen in that scenario if you've got hydrophobic,
We've done many experiments with hydrophobic surfaces coming together at exactly this level of distance, and you get this spontaneous cavitation all the time. Yeah. So we measured this. It's not made up. It's actual experiment.
Yeah. So it's based on things that are consistently demonstrated, as opposed to, models or theories that. Yeah. Okay.
So the nanobubbles are really very important, and they're missing the whole as we discussed, they're the whole missing element. The Greeks said there are 4 elements. There's fire, there's temperature, there's water, there's earth, and there's air. So we don't have air, and as soon as we got the air, all of a sudden something starts to work. And that's a part of how we got into polymers and started to get into another state of self-assembly.
Yeah, okay, because, I mean, if you're going to understand, any self-assembly process, if you like, particularly when you get into the complexity of polymers, you've got to understand the environment in which they're operating and how all of those forces are likely to interact, right? Because that's going to then be a factor in driving what assembles and how. And because assembly, or particularly complex structures, and be interested in clarification of this. The way I look at it is all these complex structures are going to form as a result of the environment in which they're faced, the salt concentration, the nanobubbles, the aminos and all those sorts of things, and their collective forces, and they're going to be stable to the degree that that environment's consistent, okay? But if that environment is now, first of all, changing those elements because, there's blood rushing past in other areas, the organism itself is physically moving around in space and therefore, shaking itself up and not only that, maybe we talk a little bit about not just the fact that dissolved gas matters, but the nature of the type of gas matters. You talked a little about, nitric oxide in particular, and that's something that certainly seems to have in the last sort of decade or so very much come to the fore about the importance and the relevance of nitric oxide. and its effects.
Yes, and it's always been, people have had difficulty with it because they try to explain it. There's something called the arginine paradox, which just doesn't fit. And in a world where dissolved gas is ignored, they don't know about it. So there's something missing in this nitric oxide problem. It doesn't make sense. But if you understand that when we breathe, the lungs are designed to form structures, open structures that contain nanobubbles and deliver the nanobubbles into the capillaries, into the salt water, so they become stable. That air that we breathe in is not just oxygen, it's nitrogen and oxygen, because nitrogen is 80% of the air, 20% oxygen. So that we breathe in nanobubbles and they get set out on their path through the capillaries, and they're nanobubbles of oxygen and nitrogen together. You don't separate them out into something. So when they get into the capillaries, they get partitioned. The oxygen goes into hemoglobin and is carried there, and that's our battery, the reserve oxygen. And then the rest of them have got oxygen and nitrogen there. Some of the nitrogen dissolved, some of the oxygen dissolved, but it's carried around and a reserve with the whole system to provide oxygen and nitrogen through directly through reactions involving glucose and through nanobubbles themselves, which have this property of producing these free radicals. So the whole thing is driven by these nanobubbles. Really.
So I just want to go back to that sort of lung surfactant sort of formation of nanobubbles being packaged in the first place. That was something we dealt with in the pulmonary surfactant COVID paper, and this, I guess, this is sort of our demonstration or imaging of this, right? So this is now looking at lung surfactant and looking at the different phases with that. So I just want to talk through a little bit about the structure. Obviously, there's a lot more detail in the actual paper, but so we've got this sort of cubic phase of lung surfactant, which is at a packaging phase. Do you just want to talk about how that actually works from us? As we breathe in, we're expanding our lung out, and our lung surfactant sort of forming a sort of single layer to cover that large surface area, and then as we expel and our lungs are contracting, we're getting this sort of phase change.
Yeah, well, this is it's been a long-standing problem. For 150 years, people have worried about oxygen gets into the system. Forget about nitrogen, they're just worried about oxygen because they know oxygen's involved in metabolism with glucose to form ATP. But so they forget about the oxygen, and then you breathe in and you breathe out. So you're breathing in nitrogen, well, where's it gone? But it turns out, and this has been known for 25 years from work that I and colleagues did, that before that people thought that in the lungs, in the lining, there's something called lung surfactant. Lung surfactant is made up of proteins and predominantly two kinds of lipids. Lipids are these cell membrane molecules that form bilayers, and the one in the lungs is called phosphatidyl palmitate, and it's got two chains, the molecule, and it's got CH2, 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 16 chains, and it's got a head group of phosphatidylcholine. And it forms a bilayer, a very rigid bilayer, and it's unique to lungs because it's the only place where you've got just two lipids that form the structure, whereas in cell membranes there's many, many of these lipids. So it forms a bilayer, and they formed a multi-bilayer, and they called it tubular myelin, and they thought this was the structure that formed in the lungs and somehow or other delivered the air to the capillaries. But it didn't work at all, and they didn't have the right kind of resolution with their electron microscopes. So it remained a mystery, and then we came along and actually observed what's going along. And it turns out it forms a honeycomb-like structure, a regular honeycomb-like structure, connected tubes, if you like, containing air and nitrogen. And these are a complex structure. You can see it there in some slight. I don't know if we've got more detail. Anyway, and it's got 4 particular proteins involved as well. It's really complex, and it forms when you breathe in. It formed this structure, and then once you breathe it in, when you breathe out, it delivers the air to the capillaries in these nanobubbles, these structures, and then as you expand again, it opens up into a bunch of silly bilayers that open up, and they spit out carbon dioxide along with water. So the breathing process is very complex. It involves something like this complicated thing there, which is when you breathe it in, and
so this is sort of this sort of, I guess, honeycomb you're sort of referring to, which is, so you're forming these sort of nanobubbles of nitrogen, oxygen that are encapsulated in these 2 lipids, surrounded obviously by water and proteins to help form that sort of self-assembled structure that's compartmentalizing the oxygen and nitrogen into nanobubbles and that's the sort of diffusion, if you like, into the bloodstream. It's not diffusion, it's containerization, another form of micellization, if you like. It's organising the gas into particles. They're actually bigger than nanobubbles, but when they're delivered to the cells on which the surfactant sits, they're broken down into nanobubbles and go into the capillaries, and they're stabilised because the salt concentration in the blood is this critical concentration, which is enough to stabilise them. Otherwise, they wouldn't be stable. So rather than diffusing oxygen through this sort of, lipid layer or monolayer in terms of the old theory, if you like, is what you're saying is the lung surfactant is there to containerise these bubbles.
That's right.
And it's almost like, reversing them through the sinter, right? To sort of say, push them into the bloodstream. Yep. And then as a result of that, going through the ESL, GC sort of into the cell, you can form the nanobubbles that will remain stable because of this salt concentration in the blood, which is a very efficient way of transporting large amounts of gas, at least from an energetic perspective, into the right size, shape, containerization, if you like, that's ultimately needed for use, in the body.
Well, with the fish, they've been around a lot longer. They have to keep swimming because they've got the water containing nanobubbles, and
they just take them straight in.
Take in. So this is a much more difficult exercise to come out on the land, which we've engaged.
Right. So that's a great contrast, if you like. And so the gill essentially is just simply allowing the already nanobubble containerized straight into and so a much more efficient system than having to sort of. But by the same token, they don't have that. There's always a constant supply of nanobubbles because there's the next bit of water, whereas, from our perspective, we're in a gaseous environment, if you like.
Well, it's not for freshwater fish. They've got to do something else too to make them more complicated gills.
Sure, yeah. Which is, I guess, also why you can't switch fish from one environment to the other, because the architecture's not there to do so. Yep. And when we have things like, those that have returned to the sea, like your whales, etc., or even deep diving seals, some of those sorts of, they have this same sort of phenomenon. They're using, containerization of oxygen as opposed to gills to be able to do that.
Yeah. Anyway, so that's
All right. So I guess, in summary, from a nanobubble perspective, is that we want to be clear that, particularly what we're referring to in terms of size of nanobubbles, right? Because that's often, I think, one of the main things that people sort of misunderstand or conflate in terms of bubbles below a certain size of nano, but there really is this separation between nano to micro to sort of macro.
Yeah, well, these are bigger than nano, but they exist. They can exist because they're containerized.
Because they're containerized,
by lipid bilayers. Otherwise, they wouldn't exist. They'd collapse. But then, as soon as they're delivered through the what are they called, epithelial cells, they're into that environment that's got that salt concentration that makes them stable nanobubbles, and off they go to do their work.
Yeah. And so, and you talked about also the energetic effect and the free energy, if you like, that the cavitation effect can create as a result of harnessing, lots of forces that are demonstrable and naturally, are there in those environments, and you demonstrate in many different ways, as an example of how the use of dissolved gas has been, how nature or biology has co-opted those forces to be able to do, very important reactions, if you like, at a rate that would just, would make life impossible without, sort of thing. So they are a fundamental component, even though they're not used in any way. So the reason an enzyme concentration, if you like, and there's many examples of where, if there's a deficiency in a particular enzyme, if you add the enzyme, everything sort of gets okay to a point, but adding more of the enzyme doesn't. It's not a dose-dependent response. It's more of a sort of threshold thing. But obviously, gas is and the amount of gas and the having the right types and availability of gas is critical. And we know that simply by, if you don't breathe and the gas isn't there, it's not just a case of there isn't the oxygen for that, for the Krebs cycle to create ATP through that chemical reaction. There's also the fact that there's all these other activities that nanobubbles are responsible for.
Yeah, well, a lot of, you mentioned the Krebs cycle and the ion pump. All of those concepts are absolutely fundamental in biology, but they're constructed from a physical chemistry that ignored dissolved gas. So you get very strange things associated with those classical theories that don't make much sense, and it's due to the fact that they're missing this concept of dissolved gas.
All right. Well, we might get into those ones next time, because that's a number of discussion in itself. But yeah, that to me provides a really sort of relatively simple, it's obviously quite a complex area, but relatively simple understanding of what are nanobubbles, why are they important in biology, how do they form, exist and maintain, and what's their, some of, obviously there's many other things we'll explore, but some of their uses. So
yeah, I think it's it's sort of it's a real revolution really in thinking, and I think it's, as I use occasionally, I think it's a bit like the ancient Greeks that thought the sun went over the sky and he went down in the evening and went to Hades and came up the next morning, and there's no question about that, it does. And so they built a whole theology and theory based on the sun going down in the evening and getting up in the morning. It's absolutely true, it does. However, it's missing something a bit like dissolved gas, that the world isn't flat, it's round, and when it's round it becomes...
But when you can't falsify that, if you can't observe the world, other than flat, then
if it's flat,
then
you've
it's
got
very
things
difficult
like
to falsify.
a lot of things in biology that are really a bit inconsistent and don't make much sense at all, and we get used to them. Forget about nitrogen doesn't exist. Forget about the fuzzy layer, it doesn't exist. And that's all consistent if the sun gets up in the morning
Comes
and
up
goes
every
out
day.
at night. But if it doesn't, well, you've got a bit more flexibility to think about things.
All right, well, we might leave nanobubbles for now, and we'll get into those next topics.
Good, thank you.